So first type of the capillary is continuous capillary, what is also called as a non-fenestrated capillary. They are a continuous lining of the thin endothelial cells. These are present in the skeletal muscles, connective tissue, smooth muscles, and lungs. They are absent in the cerebral blood vessels. So there is a blood-brain barrier is present. This is because of that the blood-brain barrier is present, and the clefts are the 4 to 10 nanometer in diameter. If we see the structure of this continuous capillary is like this. Longitudinal section and the cross section. Very small clefts are present, no gap between the endothelial cells. Second type of the capillaries are the fenestrated capillaries. They are in endothelial cells are perforated by many fenestrations or the pores. There are the many fenestrations or the pores present in between the endothelial cells. And clefts are present of the 20 to 100 nanometer in diameter. Cytoplasm of these cells is attenuated from which forms the gap, and this gap is called as the fenestrations. So type of the capillaries are fenestrated capillaries. These are present in the epithelia, as in the epithelium of the intestine villi, choroid plexus of the ventricles in the brain, ciliary processes of the eyes, exocrine glands, and parts of the kidneys. So this is the second type of the capillaries, fenestrated capillaries. See, the pores are present there. So these are called as the fenestrated capillaries. Then third one is the sinusoidal or the discontinuous capillaries. Gap is more than 400 nanometer in diameter. So there is a large transport of the large particles or large molecules occurs through these capillaries present in the liver, spleen, and the bone marrow. These are called as the sinusoidal capillaries. Large fenestrae is present. So this is the diagram of the capillaries, sinusoidal capillaries, gap between endothelial cells, which is more than 400 nanometer in diameter. Then capillary flow. The capillary flow is a different in the different tissues because there is a different pressure gradient is there in the different tissues. So flow is different for the different tissues. Then if we see the pressure in the capillary pressure at the arteriolar end, it is a 37 mm of Hg. At the venular end, it is a 17 mm of Hg. Pulse pressure is a 5 mm and pulse pressure at the venular end is a 0. Arteriolar end is a 5 and the venular end is a 0. So very less. So blood flow is a very slow due to the large cross-sectional area, and this pulse pressure is less in the capillaries. So capillary blood flow is a less pulsatile. So capillary blood flow is a less pulsatile because of the large cross-sectional area. Our pulse pressure is a very slow. Functional specialties of the capillaries, if you see, first one is the capillary density, second is the capillary diameter. Then they not contain the smooth muscles, or there is no lining of the smooth muscle. Precapillary sphincter are present at the beginning of the capillary. These are also called as exchange vessels. Least innervation by the sympathetic. There is a critical closing pressure, vasomotion, and the trancytosis. We will see one by one. So capillary density determines the degree of the exchange. So they are more in the metabolically active tissues. I told you that is in the cardiac tissue, capillary density is more as compared to the subcutaneous tissue or the cartilage. In the cardiac tissue and the glands, density is more. And less metabolically active tissues like the subcutaneous tissue and cartilage, density is the less. Second is the diameter of the capillary. Capillary diameter at the arteriolar end is a 5 micrometer and the venular end is a 8 micrometer. So at the venular end, capillary diameter is more than the arteriolar end. The diameter is very slow. What is the diameter of the RBCs? 7.2 micrometer in diameter. And capillary diameter as arteriolar end is a 5. Then how the RBCs will flow or pass through these capillaries? Blood is flowing, so RBC is also flowing. If the diameter is so less, that is a 5 micrometer at the arteriolar end, and diameter of the RBC is 7, then they pass by squeezing. They pass by the squeezing through this the lumen of the small capillaries. There is a no smooth muscle lining at the capillaries, so there is a no effect of the vasoconstriction or the vasodilators. They are not response to the vasoconstriction or the vasodilators. because no smooth muscles are present. Capillary sphincters are present. That is a precapillary sphincters. Lining of a smooth muscle is present at the beginning of the capillaries. It is called as a precapillary sphincters. And though these are not innervated by the sympathetics, because of this precapillary sphincter, they respond to the vasodilators or the vasoconstrictors. Contraction and the relaxation of this sphincter is by the presence of this sphincter. Then they are lined by the single layer of the endothelial cells. There is no muscle cell, no smooth muscle is there. So highly permeable. Thin layer, single layer of the endothelial cells. So this layer of the capillary is a highly permeable. So there is an easy and fast exchange of the nutrients and the metabolic waste products. So these vessels are called as the exchange vessels. They are least innervated by the sympathetic fibers and no smooth muscles. So sympathetic stimulation does not directly change the caliber of the capillaries. They are not directly change the caliber. Critical closing pressure, capillary pressure decreases below the tissue pressure. If the capillary pressure decreases below the tissue pressure, capillaries will be collapsed. Capillaries will be collapsed and flow of the capillaries stops. So this is called as a critical closing pressure. This is called as a critical closing pressure. Thoroughfare vessels, there is a when we see the structure of the capillaries, there is nutrient artery, meta-arteriole, nutrient artery, arteriole, meta-arteriole, and this meta-arteriole is connected to the capillaries and this capillaries to the venules. But sometimes meta-arteriole are directly connected to the venules. These vessels are called as a thoroughfare vessels. Vasomotion, flow of the blood flow through these capillaries is by the vasomotion due to intermittent contraction and the relaxation of the arterioles and the meta-arterioles. This flow is called as a vasomotion flow. Resistance is less in the capillaries because of the parallel arrangement of the capillaries. Transcytosis by the endocytosis and the exocytosis through this water soluble and water molecules are transported through this transcytosis across the capillaries. Then capillary exchange takes place by the three ways. One is the diffusion, second is the vesicular transport, and third is the capillary filtration. Diffusion is the most common way for the transport of the gases, nutrients, hormones, and other substances. So diffusion of the gases like the oxygen, carbon dioxide, nutrients like the amino acids and glucose, hormones, and other substances are transported by this diffusion. In liver, sinusoidal nature of the capillaries are present. So large molecules like the proteins are diffused through the capillaries easily. And this is an important way of transport of the synthesized proteins like the fibrinogen and albumin, which enter into the circulation. So one way of the capillary exchange is the diffusion. Second is the vesicular transport. Vesicular transport by the two ways, endocytosis and the exocytosis. Endocytosis, that is the taking of the dissolved plasma proteins and removal out by the way of the exocytosis. Substances are transported in the form of the vesicles. So this is called as a vesicular transport. And this is also across the endothelial cells for transcytosis. Most important is the capillary filtration. Capillary filtration is the major route of the transport of the fluid between the blood and the interstitial tissue space. Is the major route of the transport of the fluid between the blood and the interstitial tissue space through the capillary filtration. Capillary filtration, why takes place the capillary filtration? Because there is a gradient, pressure gradients are present. There are the pressure gradient is present in the interstitial tissue space and in the capillary space. So difference between the various pressures in the intravascular fluid, that is the pressure of the blood into the capillary. Intravascular fluid is the pressure of the blood into the capillaries, and extravascular fluid, that is the interstitial tissue pressure. Interstitial tissue pressure. So there is a difference between this extravascular fluid and the intravascular fluid. So because of this pressure gradient, there is a capillary filtration to be occurs on the text case. There are the two factors which promotes the capillary filtration and two factors which oppose the capillary filtration. promotes capillary filtration are the hydrostatic pressure of the blood, and second one is the osmotic pressure of the interstitial fluid. These two factors promote capillary filtration, that is the removal of the fluid out of the capillary. Second one is the factors which oppose the filtration, that is the oncotic pressure. Oncotic pressure is the pressure exerted by the plasma proteins which are present in the blood. And second is the tissue hydrostatic pressure, that is the interstitial fluid creates the pressure, that is the tissue hydrostatic pressure. So oncotic pressure and tissue hydrostatic pressure oppose the capillary filtration. These two factors oppose the capillary filtration. The balance between these pressures determines the capillary filtration or the net filtration. The STARLING'S forces is also important for the Viva. The forces which control the capillary filtration, these forces are called as a STARLING'S forces. The scientist Alex Starling discovered this, so name of these forces are the STARLING'S forces, which balance between the, which controls the capillary filtration. These forces are hydrostatic pressure and the osmotic pressure, or the hydrostatic forces and the osmotic forces are called as the STARLING'S forces. Hydrostatic pressure across the capillary membrane contribute the rate of the filtration. Hydrostatic and osmotic pressure difference or the gradient across the capillary membrane contribute the rate of the filtration. So pressure gradients across the capillary wall. One is the hydrostatic pressure gradient. Hydrostatic pressure gradient, which pressures are the difference between the vascular compartment and the interstitial tissue compartment. First is the hydrostatic pressure. This is the pressure exerted by the blood on the vessel wall. It is a different at the arterial end and it is a different at the venular end. Pressure exerted by the blood on the vascular wall or the vessel wall is called as a hydrostatic pressure. At the arterial end, it is a 37 mm of Hg. At the venular end, it is a 17 mm of the Hg. So difference between vascular compartment and interstitial fluid compartment. This pressure is also in the interstitial tissue. So difference between these two creates the gradient. This hydrostatic pressure in the vascular compartment always tends to pull the fluid out of the capillary. Hydrostatic pressure, it always tends to pull the fluid out of the capillary. And hydrostatic pressure gradient favors filtration both at the arteriolar end and the venular end because it always pulls fluid out of the capillary. And the arteriolar end gradient is 36 because interstitial tissue this osmotic pressure at the interstitial tissue is very negligible. It is 1. It is a very negligible. It is a 1. So at the arteriolar end, capillary hydrostatic pressure is a 37. So minus 1 is a 37. At the venular end, it is a 37. But minus 1 is a 60. So it is a more at the arteriolar end and it is a very less at the venular end. We see the diagram, see at the arteriole and the venule. This capillary pressure at the arteriolar end 37. The arrow shows full, that is fluid is always push out. Tendency to push the out. That is the arrow indicates. Maximum amount at the end and less at the venular end. Then at the venular end it is a 17 mm of Hg. Net filtration pressure is a 25 mm of Hg. Interstitial fluid pressure is very less, 1 mm of the Hg. Oncotic pressure is negligible, that is 0. Oncotic pressure is a 0 mm of the Hg. And there is an absorption because of this interstitial fluid pressure or this is the oncotic pressure. So if we see the net pressure gradient, that arteriolar end is 11. How? 37 minus 1 minus 25. It is a 11. At the venular end, it is a 9. 25 minus 17 minus 1. It is a 9. So at the arteriolar end, outward pressure direction is a 11, and at venular end, inward pressure direction, that is absorption, that is a 9. That is a 2 more in the outward direction. At the arteriolar end is a 2 plus, which is a outward. So osmotic pressure gradient, that is the difference between the oncotic pressure and the osmotic pressure in the interstitial tissue space. Oncotic pressure is a 25, interstitial tissue space is a very less, that is the 0. Osmotic pressure of the fluid compartment prevents escape of the fluid. It opposing capillary filtration. Hydrostatic pressure favors or promotes capillary filtration. But osmotic pressure opposes. It helps the fluid, or there is a retention of the fluid. It prevents. Osmotic pressure of the tissue space, the gradient on the capillary wall is always onward, which favors absorption of the fluid from the interstitial tissue space into the capillaries. Net filtration of the fluid along the capillary wall depends upon the difference between the hydrostatic pressure and the pressure gradient at the attenuated end and the venular end, which we discussed. That is, at the attenuated end, it is a 11, and at the venular end, it is a 9. So there is a difference of the 2 mm of each. Also, this along with this pressure gradients, capillary permeability also plays a role in the capillary filtration. In inflammatory conditions, there is an increase the capillary permeability. So there is a oozing of the fluid, or the removal of the fluid from the capillary to the interstitial space. So when there is an injury, that is, there is an inflammation. What happens during the time of the injury? There is a swelling, or there is a formation of the edema. Why? Increase the capillary permeability, and there is a removal of the fluid out of the capillary into the interstitial tissue space, and there is a collection of the fluid into the tissue space, and it causes the swelling or the edema. Also, there are the short vascular channels which connect directly arterioles to the venules. These are called as arteriovenous anastomosis. These are also called as arteriovenous anastomosis. They are present in the palms, fingers, and earlobes, area anastomosis. Then edema is the short one. That is the accumulation of the excess fluid into the interstitial tissue space is called as the edema. Accumulation of the excess tissue fluid into the interstitial tissue space. Excess fluid into the interstitial space is called as the edema. There are the two mechanisms for the formation of the edema. One is the increase the filtration of the fluid into the interstitial tissue space from the capillary. Increase the capillary filtration. Second one is the decrease the removal of the fluid from the interstitial tissue space. Fluid remains there. That is which factors? Now there are these two pressures are important: hydrostatic pressure and the osmotic pressure. So increase in the hydrostatic pressure, increase the secretions. So there is accumulation of the fluid. And osmotic pressure, what they cause? They decrease or they prevent the removal of the fluid. They oppose capillary filtration. Hydrostatic pressure promotes capillary filtration. Osmotic pressure opposes capillary filtration. So that mechanism is work here. So increased capillary filtration of the fluid in the interstitial tissue space increases edema, or decrease removal of the fluid. Osmotic pressure, which prevents the removal of the fluid, causes the edema. Increased filtration of the fluid in the interstitial tissue space because of by the three mechanisms. One is the increase the hydrostatic pressure of the capillary blood. Increase in hydrostatic pressure of the capillary blood. At the arteriolar end, or the venular end, hydrostatic pressure will be increases. Second, decrease oncotic pressure. That is the pressure of the plasma proteins in the blood reduce. And third one is the increase the capillary permeability. Because of the inflammation, capillary permeability will be increased. So these three factors causes the increased filtration in the interstitial tissue space. First one is the increased hydrostatic pressure. So where the hydrostatic pressure in the capillaries increases, when venous pressure will be increased, hydrostatic pressure of the venular end will be increased. So in which condition venous pressure will be increased? Congestive cardiac failure. Congestive cardiac failure, that is a decrease the pumping capacity of the heart. This pumping capacity of the heart. So there is a congestion of the blood at the veins. There is a congestion of the blood. So there is a dilatation of the veins. Finish the cardiovascular system examination. When we see the dilatation of the veins at the neck, or the distension of the neck at the veins. Why? There is an involvement of the veins, or there is a congestion of the veins. Is it because of the congestive cardiac failure? increase the hydrostatic pressure, because increase the venous pressure and causes the congestive cardiac failure. Second one is the venular constriction. Suppose there is any swelling or the tumor is present at the vein. What is causes constriction of the vein? Pressure on the vein. And because of this pressure or the constriction of the vein, it causes the drainage of the fluid. Causes the drainage of the fluid from the capillary into the interstitial tissue space. So there is a collection of the again fluid. Third one is the increased extravascular fluid volume in the fluid retention. In fluid retention, if there is an imbalance or if there is any pathology at the excretion or the kidney excretion, there is a retention of fluid in the interstitial tissue space, and which causes the swelling or the edema. Arteriolar dilatation. Arteriolar dilatation in local inflammation. Because of the increase the permeability of the capillary membrane, there is a collection of the fluid at the interstitial tissue space. So these are the causes which increase the hydrostatic pressure of the capillaries. Second, decrease the oncotic pressure. That is the decrease the plasma proteins in the blood. In diseases like the kidney diseases, where there are excretion of the plasma proteins through the urine, albuminuria. Albumins are present in the urine. It is called as a albuminuria. In the nephrotic syndrome or in the kidney diseases. So there is a removal of the proteins from the body. So decrease the amount of the proteins in blood. So there is a decrease in the oncotic pressure. Second is the malnutrition. Less intake or decrease the intake of the proteins through the diet. So it is in the malnutrition, decrease the amount of the proteins. Burns. In cases of the burns, there are the formation of the vesicles on the skin, and then the vesicles will be burst and fluid will be removed, and which contains the proteins. So there is an exudation of the protein-rich fluid from the burn areas or from the burn surface. So there is a loss of the proteins. But this decreases the oncotic pressure of the blood. Next is the increased capillary filtration. Capillary filtration increases by the action of the chemical substances like the substance P, histamine, bradykinin, and bacterial toxins. So edema occurs in the inflammation due to the increased permeability. Capillary permeability is increased. But why the capillary permeability will be increased? Because there is an increase the substances like substance P, histamine, bradykinin, or the bacterial toxins, and these substances causes the increased capillary permeability, causes collection of fluid in the interstitial tissue space. Then decrease removal of the fluid from the interstitial tissue space. Interstitial tissue space removed water by the lymphatics. Lymphatics drain out these waters. So decrease the lymphatic drainage or the lymphatic secretion causes the edema formation. Due to diseases of the lymphatics, lymphangitis, that is called as a lymphangitis, or in the radical mastectomy, that is the removal, radical mastectomy, that is the removal of the breast organ, breast organ removes in cases of the CA breast. That time there is the obliteration, or there is also the removal of the lymphatic duct, which causes the edema of the hand. And in infections like the filariasis, there is the lymphatic edema. Decreased lymph flow decreases the removal of the excess fluid from the interstitial tissue space, causes the edema. And edema, because of the lymphatic obstruction, is type of the non-pitting type of edema. We learned two types of the edema. One is the pitting edema, and other one is the non-pitting edema. If we see the edema in the nephrotic syndrome or the congestive cardiac failure, that edema is a pitting type. There is a pit will be formed, or depression will be formed if we give the pressure for the 30 seconds. In cases of the lymphatic obstruction, edema is present, but it is a non-pit one. There is a pit will be not formed, no depression will be formed. What is the treatment for the edema? For the generalized edema, administration of the diuretics, that is the increase the excretion of urine, increase the urinary output. Diuretics are the drugs which increase the urine output. Second, restriction of the salt restriction, because salt causes the retention of the fluid in our body. So salt restricted diet is advised and diuretics are given, increase the urine output. Second, for the localized edema, which causes because of the inflammation, anti-inflammatory drugs are given. Anti-inflammatory drugs are given for the localized edema or edema due to the inflammation. Then special form of the edema, diuretics or other drugs depending on the cause of the edema. Suppose in the congestive cardiac failure. In congestive cardiac failure, there is a cardiac output will be reduced, or there is decrease the function of the heart. So what we do? The treatment which increase the cardiac function, digitized drug is given in the congestive cardiac failure. Like this. So depending upon the cause of the edema, treatment is the given. Then vascular response to the injury. There are the two types of the responses occur. One is the white reaction and second one is the triple reaction. One is the white reaction and second one is the triple reaction. Triple reaction is an important short form. So white reaction, if we stroke the skin with the light object or the lightly, suppose you take the pen and stroke your skin lightly, what happens? White line, you see only the white line is there. It is called as a white reaction. If we stroke the skin with the pointed object and deeply, what happens? Injury will be there. Red line will be occur and it shows three reactions. It is called as a triple reaction. There it shows the three reaction. It is called as a triple reaction. So first is a white reaction. White reaction when skin is a stroke lightly with the pointed object, stroke light becomes pale or white line. We can see the white line. It is called as a white response. It is because decrease the blood flow in the capillaries due to contraction of the precapillary sphincter. Why there is a white reaction? We can see it is because of the decrease the capillary blood flow to that area. And why capillary blood flow is decreased? Because of the constriction of the precapillary sphincter. So no blood flow is there. So no red reaction is there. Only there is a white reaction, and this is observed within the 15 seconds. This reaction is occurs in the 15 seconds. This is called as a white reaction. Second is a triple response. Second one is triple response. When skin is stroked with a pointed object. Stroked with a pointed object. The response is called triple response as it has three components. Why it is called as a triple response? Because this response has the three components: red reaction, wave, and flare. Three types of the reactions are there. Red reaction, wave is there, and flare is there. So first is the red reaction. After stroking with the pointed object deeply, red line is seen within the 10 seconds. Skin becomes red. After stroking within the 10 seconds, skin becomes red, a red line is there. It is called as the red reaction. Why there is a red reaction? Because of the capillary dilatation. Increase the blood flow to that area. Because of capillary dilatation, blood flow increases and it is a direct response of capillaries to the pressure which will be exerted by the stroke. So there is a dilatation of the capillary, increase the blood flow, and we got there the red line or the red reaction. Second is the venule, or it is also called as the swelling or the edema. Local edema is there. Is called venule. Within the few minutes after red reaction, occurs due to increased permeability of the capillaries and post-capillary venules. Now capillary permeability will be increased. So oozing of the fluid out of the capillaries into the interstitial tissue space, and there is a formation of the swelling or the edema. It is called as a venule, and this is because of the release of the histamine from the local mast cells. Which cells releases the histamine? Mast cells. So there is a release of the histamine from the local mast cells, which causes vasodilation and increase the permeability that results in extravasation of the fluid. And oozing of the fluid from the capillary into the space, and collection of the fluid into the space causes the edema. It's called as a venule. So see, this is the triple response. Stroking with the pointed pencil First we see there the red line, and second there is a vein, and then there is a flare. Why occurs there? See, red line it is on the skin, that is on the dermis, and that is at epidermis, and what happens in the dermis. Nerve endings, pain sensitive nerve endings are there. There is a stimulation to the brain. There is a stimulation, impulses are goes to the brain, and then there is a release of the substances like the histamine. And there is a accumulation of the fluid. So this is the diagram of the field response. Third one is the flare, spreading. There is that is a spreading of the redness from the site of the injury to the localized area of the surrounding area. How redness will be flares. It is called as a flare. It is because of the arteriolar dilatation. Cause of everything is different. First, there is white reaction because of the constriction of the precapillary sphincter. Second, red reaction because of the capillary dilatation. Then there is a vein because of the releases of the histamine. And there is a oozing of the fluid. Third, red flare is there. It is because of the arteriolar dilatation and activation of the axon reflex. Axon reflex. If there is a short note on axon reflex, also you write this axon reflex. There is a arteriolar dilatation occurs because of the activation of the axon reflex from the site of injury. Impulse conducted through the afferent fibers, and these sensory neurons gives branches to the blood vessels. The impulse in addition to its conduction to the spinal cord orthodromically is also relayed antidromically to the blood vessels. Conduction of impulses from the skin to the brain, also to the blood vessels. It is an antidromical. Axon reflex is an example of the antidromic conduction of the impulse. Is the example of the antidromic conduction of the impulse. Endings of the sensory fibers of the blood vessels releases substance P, which causes arteriolar dilatation. Thus, when there is an injury, there is a surrounding area of the skin is called the flare. So this is the diagram of the axon reflex. When you are stroking the skin on the cutaneous nerve endings, impulses are goes through the spinal cord to the brain. Also, the impulses go to the skin arteriole. So there is a dilatation of the artery. It is an example of the axon reflex or the orthodromic conduction of the impulse. Vascular response to temporary occlusion. If there is a temporary occlusion, what happens there? Reactive hyperemia is there. That is increased blood flow to that area. Increased blood flow to that area. When blood supply to the area is re-established following a brief period of the occlusion. It occurs due to vasodilatation produced by hypoxia during occlusion. When circulation is re-established, blood flow increases through dilated vessels and skin becomes red. So it is because of the reactive hyperemia. Because of the hypoxia, there is a vasodilatation will be occurs. So this is the capillary circulation. Two short notes. One is the triple response and second is the edema.